actually like, I don't know why I put jokes here. This is not time for jokes.
Are you serious?
Yeah.
All right. Anyway, I like my jokes.
I like them too.
I know.
So, just in terms of the history of genetics, you guys know about Mendel from 7th grade. He found out basically the main statistical breakdown of how organisms reproduce and pass on those traits. He was using pea plants, had very meticulous notes, and did thousands and thousands of crosses between plants to see the pattern of inheritance. And it was actually kind of lucky he used pea plants because pea plants exhibit pretty clear traits that are dominant or recessive. Most those traits are not linked, so he sort of picked an ideal plant to do his experiments with. And there's lots of different factors that Mendel looked at, including the shape of the seeds, the color of the seeds, color of the flowers, the height of the plants, and so forth. And he found some patterns. He found that when he crossed these plants in different configurations, they always ended up with similar ratios of dominant to recessive traits. And that allowed him to make his theories about how these traits are inherited from parents to their offspring. Now, I should say, and we'll talk more about this probably tomorrow, that human genetics and genetics of most organs are much more complex. There really are very few traits that are just a simple dominant, recessive, that are inherited in these very clear patterns. Many traits of most organisms are controlled by multiple alleles and multiple genes, and the inheritance is much more complex than kind of we make it sound. So these are some simplified rules that apply in certain situations, but also they don't really apply in other situations because there's more complexity. You guys watch that video or no?
No.
Okay.
Can we watch it?
No. So Mendel's laws are sort of the three laws that Mendel arrived at after doing his experiments. And we'll just mention those here. So the first one, the law of dominance, is something you're familiar with from 7th grade, in that in certain traits, there are some alleles, we call them now. We didn't use that word in 7th grade, but some alleles, some versions that we call dominant, that exhibit sort of more strength when they are inherited. And when one of these dominant alleles is present, that is what shows up in the offspring. That's what's presented in the phenotype. Okay? So the dominant sort of, you can think of it as covers up. And the symbol we use for dominance is a capital letter. We use the same letter, but lowercase version generally for the recessive allele. And so when we look at Mendel's work that he did, what he found is that if he took pure tall plants, so plants that had two of the tall alleles, two big T's, and crossed them with pure short plants, plants with two of the recessive alleles, what he found is that the offspring 100% of the time were tall. There were no short plants ever represented when he crossed a pure dominant and a pure recessive plant. So these terms, I think you know, the genotype are the genes that the organism has. The phenotype is the expression of those genes. Now, here I said physical characteristics, but it doesn't have to be like an outward appearance sort of thing. Our genes control also how our body works, how our enzymes are produced. So it's sort of like the physical manifestation of the genes. But it doesn't have to be some outward appearance. It can also be other things that are important internally. Homozygous means two of the same alleles. Heterozygous means one of each. So the parent generation, the P generation, are the original parents. So when Mendel did his work, his parent generations, they were pure tall plants and pure short plants. And when he crossed them, 100% of the offspring, the F1 generation, the first generation of offspring, they were all hybrids. Okay? They were 100% heterozygous. They were 100% tall phenotypes. Then Mendel would take these plants and then cross those together. Take two of these F1 generation to produce the next generation, the F2 generation. And what Mendel found is when he crossed this F2 generation, these heterozygous plants, he found that they always produced the same ratio, okay, That 75% of the offspring were tall, and 25% of the offspring were short. When we do the Punnett square, we see that's what we would expect. Okay? That if you cross 100 plants, produce 100 offspring, about 75% of them are going to be tall, and 25% are going to be short. So that's Mendel's first law, the law of dominance. That this dominant shows up, sort of covers up the recessive gene when they're inherited. Then we have Mendel's other two laws, the law of segregation and recombination. Law of segregation and recombination tells us that when diploid cells, cells that still have their chromosomes in pairs, each homologous chromosome is carrying one allele. Okay. So we have our plants here, our generation here, our F1 generation, the hybrids, the heterozygous plants. And when these diploid cells produce haploid cells during meiosis, when they produce gametes, remember those homologous chromosomes separate. Okay. And they each end up in their own gametes. So this cell that has the dominant gene, dominant allele, and the recessive allele, when it produces gametes, well, some of the gametes will get the dominant allele, some will get the recessive allele, half and half. Same thing happens over here. So that's segregation, that these genes, these alleles, get separated when gametes are being produced because of meiosis. After those gametes are produced, when they recombine during fertilization, then we go back to our normal chromosome number, and we have the recombination during fertilization of those gametes to go back to 2 of each alleles. And that happens randomly when a sperm cell fertilizes an egg cell or pollen fertilizes an ovule in a plant. That's sort of random. And so which gametes fertilize determines the genotypes of the offspring. And then the last law is the law of independent assortment. This means that the various traits are inherited independently of each other. So when Mendel was studying pea plants, he saw that whether or not the flower was purple was not related to whether or not the seed was yellow or green, which was also not linked to whether it was smooth or wrinkled. That each of these traits were inherited separately from each other. Okay? And that's independent assortment. Okay? And that happens because we know these alleles that Mendel was studying, they were each on a separate pair of chromosomes. And so when those homologous chromosomes separated and formed the gametes, right, they all were inherited independently of each other. We'll talk about sometimes when they are linked in a minute. Because when gametes are produced, we know how many chromosomes in a human? 46. They align themselves in 23 pairs. The pairs separate and each end up in their own gametes. But sometimes the alleles, which are, so this is a chromosome. And on a chromosome there are thousands of areas that contain the genetic information. That's where the genes are. They're in the DNA, in the pattern of bases. And so one chromosome has many, many genes on it. Okay? They've been mapped over time by scientists studying. So scientists know that the gene for eye color is on a certain chromosome. They know where on the chromosome it's actually located. Okay? And so this has been mapped. Okay? And what this means is that if the gene for a trait, 2 separate traits, are on the same chromosome, and they're close together, they are probably going to be linked together and inherited together because they're on the same chromosome. So if we imagine this chromosome here on the left, see these gene 1 and gene 2. They're very close together on the same chromosome. So if this chromosome ends up in a gamete that produces an offspring, probably these 2 genes are going to go together. They're inherited together. They are what we call linked. What could break that linkage? So if they're on the same chromosome and that chromosome ends up in a gamete, all the traits that are on that chromosome probably are going to be inherited together. Caitlin? Yeah, the time when those linkages can be broken is when the chromosomes trade pieces, during crossing over. Because now, if these two genes instead were if these genes are far apart on this chromosome, then crossing over, this part of the gene could be exchanged with another chromosome, and now gene 1 and 2 are no longer linked, because crossing over has put those on separate chromosomes. If they're very close together, the chances of crossing over, sort of splitting those two traits, is very low, because they're so close together. The farther apart they are, the more likely that linkage can be broken. And that is actually how scientists have mapped genes to specific chromosomes. They look and they can see, okay, how often is red hair and freckles, how often are they inherited together? What do you think the answer to that is?
Pretty often.
Very often. So what does that mean in terms of the chromosomes? It means the genes that are responsible for red hair and freckles are very close together on the same chromosome. So if you inherit that chromosome, most likely you got the gene for red hair and you got the gene for freckles and the gene for light eyes. They are linked because they're close together on those chromosomes. Now if they happen to be far apart, crossing over could make it more likely that those get separated. And this is how scientists sort of put together these maps of the genes. They look, okay, how often are these traits inherited together? The more often they're inherited together, the closer they are on that gene. So that's how these are put together. So we talked earlier today about a test cross. Again, when we see that the dominant phenotype is present, like purple flowers, we don't know if that's because it's a homozygous purple or if it's heterozygous. Both result in purple flowers. So we do our test cross, and we cross this plant with a plant that is homozygous recessive, has two of the recessive genes. If the original plant was homozygous dominant, we're going to find all of the offspring purple 100% of the time. If the original plant that we're studying was heterozygous, we're going to expect that 50% of the time, okay, it's going to be purple, and the other 50% is going to be white. So that's what a test cross does. It tells us about the parent. Okay, we could study, we don't need to study just single traits, like those simple Punnett squares. We can study multiple traits at the same time. We could do 2, we can even do 3. Beyond 3, it gets sort of unwieldy to do by hand. But let's say, why is that there? I want to be careful. If we have, let's look at this example on the left first. We're going to look at 2 traits at once. Okay? We're going to look at a hybrid long plant, long being the dominant gene. So it's hybrid long, so big L, little l, crossed with a pure wrinkled plant. So pure wrinkled is little r, little r. So we know that's the parent genotype. We're going to cross this plant with a short plant that's also hybrid for round. So short plant would have 2 little l's, and a hybrid round means a big R and a little r. And so when we have these multiple traits at one time, we have to look at, okay, what are our possible combinations that we can have? So if we look first at this parent. So which gene can they get for long or short? Well, there's only one option, right? There's only 2 L's. So the first letter is definitely going to be a little L. There's no other allele that they could inherit. When we look at the allele for wrinkled versus round, well, they may get the big R, or they may get the little r. So for this plant, there's only 2 possible gametes that could be produced, and they're listed here. Little r, big R, little r, little r. If we look at this plant, what are the two possible gametes that could be produced from this parent? Big L and little r, or little l, little r. Those are the two possibilities. It can get the big L or it can get the little l, but it only has the little r that it can get. We then complete our Punnett square the same way. So we get big L from here and a little l from there, and we get big R, little r. Sebastian, what should go in this box?
Big L, little L, two little R's.
Correct. Nicola, what should go here?
Little L, big R, little R.
Correct. Alvy, how about this last box?
Two little L's and two little R's.
Correct. Okay, we look at our phenotypes in the same way. Okay. So in this case, 25% of them are going to be long and round. That's this one. 25% are going to be long and wrinkled. That's this one. 25% are going to be short and round, and then 25% short and wrinkled. That's a fairly simple example because there's only two possible gamete combinations. This one's a little more complex. Okay? So, let's start with this parent. So we have pure long crossed with a round hybrid or heterozygous. So what are the possible gametes that could be formed from this parent? Chloe? What gametes could this plant form?
Big L, big R, big L, little R.
Correct. So it's going to get a big L. That's the only possibility here. And it could get the big R, or it could get the little r. Cormac, what gametes are possible from this? This is a hybrid in both traits. So hybrid long and hybrid round.
Big L, big R, big L, little R, or little L, big R, little L, little R.
Yeah, so this one gets all the possible combinations. Could get big L, big R, could get big L, little r. Or if it gets the little L gene allele, it can get the big R, or it can get the little r. So there's 4 possible gametes that could be formed from this parent. Again, we complete our Punnett square in the same way. Matt, what do we get here?
Big L, big L, big R, big R.
Correct. And what would it be like? How many phenotype?
That would be tall and round.
Yeah, long and round. How about this one, Dan? What do we get for genotype here?
Big L, and big R, little R.
Correct. What do we get here, Antonio?
2 big L's, one big R, and one little R.
Yep. Caleb, how about here? What do we get here?
Big L, little R, little R.
How about here, Nate?
Big L, little L, big R, big R.
Yep. All right, so you guys got this?
Yeah.
All right.
Instead of saying big L, we just say big L.
All right, so when we look at the phenotypes, again, we just have to break it down by each trait. Okay? In this case, 75% of them are going to be long and round. That's all of these. Okay? And this one and this one. 25% are going to be long and wrinkled. So these are 2 trait problems. The most complex 2 trait problem we can get is what?
Hybrid.
Dihybrid cross. So when we have 2 traits and 2 of the parents are both hybrids for both of them, that gives us our maximum number of possibilities. Okay, that's what's shown here. So this is a dihybrid cross. We look closely here, okay, that we have hybrid for yellow, big Y, little y, and also hybrid for round peas, big R, little r. If we cross two of those, we end up with, each has 4 gamete combinations, okay? So we could get big Y, big R, big Y, little r. We could get little y, big R, little y, little r. And we could get the same for the other parent as well. So there's 4 possible gametes for each parent. We complete our Punnett square in the exact same way, okay? We get all 16 of these combinations, okay? And we can look at how many out of the 16 have the various phenotypes, yellow round, green round, and so forth. But it's basically just like the traits we did, just a little bit more complex because we have a couple more possibilities. So does anyone have questions about the dihybrid cross? It's the same procedure, just you have 16 squares there. A trihybrid cross gets quite complex because, again, every extra trait that you add just increases the complexity. So when we look at a trihybrid cross, we're looking at something that has 3 traits. Here is an example, A, B, and C. 3 separate traits that we're looking at. If the parents are hybrid for A, B, and C, okay, what that means is we end up with 8 possible gamete combinations. You could have big A, big B, big C, and so forth. You can set this up using a little tree to help you see the various gamete possibilities, okay? And so each parent has 8 possibilities. So when you do the actual Punnett square for it, you have an 8 by 8 Punnett square with 64 possible offspring. It looks complicated, but really it's the same as the dihybrid cross. It's just with more boxes to fill in. So again, the possible gametes that are produced by a trihybrid, you can see them listed across the top, and you see them listed across the side. If we do the combination, you can see these are all the offspring possibilities. You could figure it out mathematically as well. That's what this is showing. You know, when you look at the phenotypes, there's 3/4 of a chance that they're going to show the dominant phenotype. So there's 3/4 chance it's going to show the dominant trait A, and 1/4 chance it'll show the recessive. And then if we continue our tree, we can figure out the probability of it ending up with all 3 dominant genes is 3/4 times 3/4 times 3/4 for 27/64. Okay? If you want to find any other combination, you just have to multiply the probabilities. 3/4, let's say you want to know if it's going to be big A, little b, little c. Big A, 3/4 times 1/4 times 1/4, you can find the probability. So you can figure it out mathematically as well. We're not going to have to do a lot of these examples, but you could do 4. You could do 5 or 6 traits if you really needed to. Your grid just becomes bigger and bigger. You have more and more traits, but the process is the same. All right. Any questions about those things?